All right, guys, let's think about how we complete this cycle, the TCA cycle, that is so important to the generation of ATP at the highest possible level of efficiency in living organisms, right? So remember, let's just a quick review of where we've come from, right? A little accounting just to sort of set the stage. Remember, we started out with pyruvate coming out of the end of glycolysis, right? And we came down out of this pathway, or to start this pathway, we ran that pyruvate dehydrogenase reaction, right, as you guys recall, hopefully, which we used some NAD plus to make some NADH, right, to drive this decarboxylation reaction, all right, this oxidative decarboxylation, to turn pyruvate into an activated 2-carbon unit, essentially using this oxidative decarboxylation reaction to synthesize this activated thioester of acetyl-CoA, right? Right, okay. So the next thing we did, if you recall, is condense acetyl-CoA with oxaloacetate, right? So put these guys together, oops, through the citrate synthase reaction to make this tricarboxylic acid citrate, for which the cycle gets its primary name, right? And then we drove these guys in the last video. We saw how we drove this citrate through this sequential series of essentially oxidative decarboxylations, again, peeling off some more NADH, right? And driving off more CO2. But if you remember, we had to do, there were sort of 2 steps of this process, right? We went through 2 oxidation and decarboxylation steps, right? When we drove this thing through the isocitrate dehydrogenase and then the alpha ketoglutarate dehydrogenase reactions, right? So for that single reaction of taking oxaloacetate, making that C6 citrate, right? We then drove off, or I guess you could say, for one pass through this first part, my sort of phase 2 of the cycle, we did this NADH, we did that twice, right? Because we ran the isocitrate dehydrogenase and the alpha ketoglutarate dehydrogenase. So that was 2 oxidation steps. So we got 2 NADH there. And so we got 2 CO2s out of this, right? Okay. But if you remember, if you think about normalizing this to carbon equivalents, right, relative to a glucose, there's more than that. Because remember, per glucose, we have 2 pyruvates per glucose, right? Which means we did, per glucose, we did this twice here, right? So the 2 pyruvates came down the first part of this pipe, which also then means per glucose, we did that twice, which means at this point, we've gotten 4 NADHs per glucose equivalent, right? And 4 CO2s per glucose equivalent, right? Because each pyruvate, right, each acetyl-CoA equivalent, there are 2 of these that come through the cycle per glucose. Okay, you add that up, like, oh, well, that's kind of cool, right? Because that's 4 carbons, right? That's another 2 carbons. 4 plus 2 equals 6, and there are 6 carbons. That's it. That's a glucose, right? We've run a full glucose through TCA cycle at this point, right? It's only one problem. Remember, what was our product down here at the end of phase 1, right? When we came through that alpha ketoglutarate dehydrogenase reaction. Remember, we synthesized this activated thioester compound, succinyl-CoA, right? And if you recall, because of the stereospecificity of the aconitase reaction, right? Remember, at this point, I know those are the carbons that I brought in here from acetyl-CoA. You're like, wait a minute. So the carbons I brought in are still there, but I've burned a glucose equivalent of carbons. Where did the rest of those carbons come from? Well, if you think about it and you remember that this is a cycle, and the oxaloacetate, that's a 4-carbon unit that was already there, right? Essentially, when the acetyl-CoA came in, all but 2 of these carbons, 2 of those clearly came from the glucose that I just brought in, right? From that pyruvate. But the other 4 carbons that we threw off here, they have to have come from previous glucose, from glucose that came in the cycles before, right? Which means at this halfway point, essentially, the end of phase 2 of the cycle, the way I like to think about it, right? Again, reminding you guys, if the pyruvate dehydrogenase reaction, if this is phase 1, right? And the first half of the cycle following synthesis of citrate around to the formation of succinyl-CoA, if this is phase 2, we've only at this point burned through 2 of the carbons that came in from the original glucose as pyruvate, right? We got 4 more to go, essentially, because remember, there are 2 of these per glucose, 2 of these succinyl-CoAs per glucose. We got some more work to do, right? Okay. All right. So let's think about that next set of reactions. So we ended the previous reaction, if you recall, the alpha-ketoglutarate dehydrogenase reaction. That was reaction 6. So our next step forward then to enter phase 3 of the TCA cycle, we're going to call that reaction 7, right? So again, we're going to take that succinyl-CoA molecule, right? So here's our CoA, right? It's 5 carbons, 1, 2, 3, excuse me. Carbon, 1, 2, 3, 4, right? And now we're going to take this guy, we're going to take some inorganic phosphate, right? We're going to take some ADP, or some isoforms of the enzyme GDP, right? And we're going to basically use that thioester bond in succinyl-CoA. Remember, that's a high energy bond, right? That thioester bond is storing some free energy. It's unstable. We're going to use that to power the only substrate level phosphorylation reaction of the TCA cycle. We're going to basically blow up that bond, use the free energy available in that bond, which will then give us this dicarboxylic acid, succinate, plus some ATP, or again, depending on the isoform, GTP, right? So you look at that, and I should tell you the enzyme that runs this reaction is known as succinyl-CoA synthase. Again, like so many of these enzymes we've seen named for the reverse reaction, relative to the one we're thinking about running in the forward direction for TCA cycle. You look at that and you're like, wait, what did you do? Wait, you had a thioester bond, now it's gone, and you have some ATP. What gives? That didn't just magically happen, right? All right. The direct analog to this set of the reactions here should look kind of familiar to you if you remember the pyruvate dehydrogenase reaction. Remember that reaction, right? Pyruvate dehydrogenase, we ran an oxidative phosphorylation. Well, I guess the pyruvate dehydrogenase reaction followed by the subsequent reaction, right? If you remember, we used that oxidation reaction to create a thioester bond in the active site of pyruvate pyruvate dehydrogenase, GAPDH, glyceraldehyde 3-phosphate dehydrogenase. Remember, we had an aldehyde group. In the active site of the enzyme, we temporarily created a thioester bond, right? We used the free energy of that aldehyde to form that internal that hemithioacetal, oxidized it to create that thioester bond, which stored a good chunk of the free energy there, bled those electrons off into NADH, and then used that thioester bond to power the formation of that acyl phosphate group to make 1,3-bisphosphoglycerate, which we then, because of its high phosphotransfer potential, used to make ATP. Okay. If you look at this reaction, what did we do just upstream of this, right? We ran an oxidative decarboxylation. We decarboxylated in this case, but we ran an oxidation reaction using NAD to make a high energy thioester bond, right? Kicked off a CO2 in this case, didn't have an aldehyde, but kicked off that CO2 and used that oxidation reaction to make that high energy thioester bond right here, right? Okay, so now we've got that guy. Now, what we're going to do is exploit the formation of that thioester in succinyl-CoA to run a phosphorylasis reaction to make an acyl phosphate group, just like we did with the GAPDH reaction. And then in this reaction, sort of essentially 2 steps in the same reaction instead of a subsequent reaction, we're going to use that to make some ATP. So the way this works is in the active site of the enzyme, this succinyl-CoA, as I said, is used to run a phosphorylasis reaction where we bring in some inorganic phosphate to basically replace that thioester with a phosphoester bond, right? To make this succinyl phosphate compound. This never leaves the active site of the enzyme, right? So it's never a free intermediate, but it would be directly analogous to what we did with to create 1,3-bisphosphoglycerate in glycolysis, right? Where we've now split that bond to give reduced CoA, right? To now make that succinyl phosphate compound, which can then be used in a direct substrate level phosphorylation event to take ADP and make ATP, or, as I said, in some isoforms of the enzyme, take GDP to make GTP. Okay. So a couple of interesting wrinkles here. Well, look at how the enzyme does this. The actual chemistry is fairly straightforward, but the structural biology of how this enzyme works is kind of interesting. The first thing, though, before we get to that, is this issue of ATP versus GTP. Now, you often see succinyl-CoA synthase written down as making GTP when you look in textbooks. And the reason for that is because the liver isoform in organisms like us, the liver isoform of the enzyme does primarily make, it makes GTP. It uses GDP to make GTP, a large fraction of which gets turned into ATP by an enzyme called nucleoside diphosphate kinase that basically takes GTP plus ADP and makes ATP plus GDP. It basically just shuffles the phosphate group over, right? Okay, but like I said, this is mostly happening in the liver, right? And because so much of the biochemistry of many of these pathways is worked out using liver tissue, there's sort of a bias in thinking about the metabolism, a lot of this intermediary metabolism from a liver-centric point of view. Turns out the isoform of succinyl-CoA synthase in heart muscle, in brain and nervous tissue, neurons, it actually uses ATP or uses ADP, right? It doesn't use GTP. So different tissues in an organism like us will use either the GTP or GDP dependent isoform of this enzyme or the ADP dependent. Lots and lots of microorganisms and many plants also use the ADP utilizing form of this enzyme and not the GDP form. All right, so that's where some of that comes in. And like I said, because of nucleoside diphosphate kinase, you can think about this as kind of equivalent, right? But there's enough of a wrinkle there, and you see it written down that way enough, it's worth being aware of. Okay. The second thing to sort of be aware of is how this enzyme works. And this is where, if for those of you who are listening in lecture today, I screwed up, right? Like I said, you always be humble in the face of biology. Avoid the use of never and always, because, man, evolution is going to find a way around you. And I made this statement that I thought PGM, phosphoglycerate mutase in glycolysis, was the only enzymatic step that used a phosphoamide bond, and that's wrong, right? Succinyl-CoA synthase, right here, right, if I just thought about it harder, also uses a phosphoamide bond, a phosphohistidine bond, to make ATP. So I screwed up, right? But let's look at how this enzyme works, because it's kind of cool. All right, so here we are, reaction 7. We're going to go on past this little scheme. Here's succinyl-CoA synthase, right? It's a, again, I can't remember. This is an ADP-linked form of the enzyme, so I'm betting this is a bacterial isoform of the enzyme. Certainly not the liver form of succinyl-CoA synthase. It has 2 subunits, so-called alpha subunit and a beta subunit. The alpha subunit is this big guy over here, and the alpha subunit, I'm sorry, this is the beta. Yeah, no, this is the alpha subunit over here, right? Or the smaller bit over here. This sort of larger structure over here, yellow and the red, is the beta subunit. You'll notice there's a domain you've seen before, the so-called Rossmann fold, except this isn't a dehydrogenase enzyme. The Rossmann fold crops up really frequently in dehydrogenase enzymes. Here's an example of a protein using a Rossmann fold, but it's not a dehydrogenase enzyme. Instead, what it's using that Rossmann fold for is as a way to recognize the adenine component, right, of that CoA moiety that's on succinyl-CoA, right? And it's bound right up in here. Over here in the beta subunit, this sort of red part of the beta subunit, is the so-called ATP grasp. An ATP grasp protein. It's a particular type of ATP binding motif, right? It's distinct from many of the others, but it's one type of structural motif that binds ATP pretty well. And it's over here. If you notice something like, well, wait a minute, what? Don't enzymes work by proximity and oral alignment and close apposition of subunit? How does that work? You've got the succinyl-CoA over here. We got to put the phosphate on there, but the ATP's over here, ADP's over here, right? So this is the cool structural part of this. This enzyme works with kind of a swing arm, not as sophisticated as the pyruvate dehydrogenase complex or alpha-ketoglutarate dehydrogenase, but sort of a similar idea in some ways. All right, so here's the succinyl-CoA, right? Here, bring some inorganic phosphate in. We're going to run this phosphorylasis reaction to create this acyl phosphate group. And then for interesting reasons that I don't have the explanation for, once we form that acyl phosphate group, that phosphate group is then transferred to a histidine, right? That's sitting nearby, that's in a loop. It's kind of a little hard to see here, but it's right there. If you look at there's this loop, right? That's part of the beta subunit. The histidine group is right there. There's the thioester, right, of the acetyl-CoA. The phosphate group, once it gets put onto the acetyl-CoA, it then gets transferred, onto the succinyl moiety, the succinyl phosphate is created. Phosphate group is then transferred to that histidine. And when the phosphohistidine is formed, there are 2 things that happen. The succinate is released. That phosphohistidine in that loop, the structure senses that, and it goes through this fairly significant structural rearrangement. And what happens is that loop swings all the way from the active site over here between the beta subunit and the alpha subunit, and it swings all the way back over here to the ATP grasp domain over here in the beta subunit, right? So basically the phosphohistidine moves from one active site over to the other active site and transfers then that activated phosphate group over to ADP or GDP to synthesize the phosphotriester, right? Okay, so that's kind of cool. It's a neat structural wrinkle to this enzyme. That's about all I want to say about the mechanism of that enzyme. So that more or less completes reaction 7. We've made succinate. We've made our only actual phosphoester bond directly in the cycle, substrate level phosphorylation event. So let's go on and see if we can complete the cycle, right? So we'll go down here to reaction 8. All right, so coming out of the succinyl-CoA synthase reaction, we had, as I said, succinate, right? So we're already down to 4 carbons, like I pointed out before. Remember that we still got our carbon identity here because of the, at least coming in, we know that we could track those carbons coming in because of the stereospecificity of the iconotase reaction. But at this point, we're about to lose it, right? We're about to lose that carbon identity, right? Because the next enzyme, which actually has a covalently attached, I'm going to write it out this way, covalently attached flavin adenine dinucleotide. It's got an attached FAD, so oxidized but covalently attached to the enzyme. This enzyme is now going to come in here, right? And it's going to staple in between that 2 and 3 carbon in the middle of this molecule, a carbon-carbon double bond, right? Right here, to create an enzyme, a molecule known as fumarate, right? And at this point, this is a completely symmetric molecule coming out of the backside of this reaction. So we can't distinguish chemically, right? The CO2 group here and the CO2 group over here, they're chemically identical. We can't distinguish them any longer. There's no way to determine, once this molecule has been synthesized, which is which means the chemical identity between the 2 halves of the molecule is lost. So essentially, the 2 halves of this molecule are more or less exactly the same, and the subsequent steps really can't distinguish anymore, right? So if one had, say, radioactively labeled that carboxylate group, the subsequent steps would, half of that radioactive signal or the isotopically labeled signal would respond in one way, and half would respond in a different way, right? So that's something to keep in mind here. At this step, and this enzyme is, of course, known as succinate dehydrogenase. Right, and go over here, write out we're going to get some FADH2 out of this, right? The free energy change for this reaction is approximately 0, right? It's the standard state, right? The actual, again, this is one that I haven't been able to locate. It's going to be less than 0, but it's going to suffer from the same thing as so many of these steps that are operating unfavorably in terms of the standard state free energy in the forward direction. They're going to have to be pulled, right? So the subsequent steps of the cycle, ones that bring us all the way back up to oxaloacetate, are going to have to drain that fumarate pool pretty effectively in order to get this thing to move in the forward direction. And that's exactly what happens when the flux goes in this direction, right? I think I forgot. Yes, I didn't write it up here. Again, I'm not sure I have the actual numbers. I'm pretty sure I don't. The standard state free energy change for the succinyl-CoA synthase reaction, I don't even have the standard state, never mind the cellular conditions, but both of these are significantly less than 0. This is a very favorable reaction because we're burning the free energy of that thioester bond to create ATP and GTP, and the phosphotransfer potential of that acyl phosphate group there is quite high, right? So this is a very favorable reaction. All right, anyway, back to succinate dehydrogenase. So essentially what we've done here is stick that carbon-carbon double bond straight in the middle of the molecule. You could look at that and go, well, wait a minute, how does that work, right? If you think about this molecule, how on earth you have something that looks like this, right? And we've got here, we got these protons here. We got basically these methylene protons here. How on earth do you get that? That's what you got to do. You got to pull, right? You got to pull a couple of these guys. to make that put that carbon-carbon double bond in there. How do you do that, right? I mean, methylene protons are something you can normally yank on, right? They're hard to get off. Well, the answer to this, of course, is that because those carbons are alpha to that carbonyl group, in principle, they're much more acidic than a normal methylene group would be, right? You can imagine, in principle, you can imagine, say, pulling that guy here, and then you could push the electrons up there. So there's a sink, in principle, you could imagine shoving those electrons into. Now, that's not going to happen very easily, right? That double oxyanion on that carbon, that's super unfavorable, right? If you tried to do this, this would turn around in reverse and go back in the opposite direction quite readily and just put the proton back on there. But if you have some FAD nearby that's capable of taking up that proton when you destabilize it, and you do something to sort of make this more readily available, like you start pulling on it with a base, right? You can pull that proton off because it's significantly more acidic by virtue of these carbonyl groups that are nearby, and then suck the electron off at the same time because you have that nice sink and that FAD. You can actually pull both of these protons and their associated electrons into the FAD to make FADH2 in a way that you couldn't do that if you didn't have those carbonyl groups nearby, right? So those carbonyl groups make those protons and their electrons significantly more abstractable than they otherwise would be if that was just a normal methylene carbon, right? Okay, another wrinkle about succinate dehydrogenase. This is the only enzyme of the TCA cycle that's membrane bound. This is an integral membrane protein, right? Right, and what you're going to see, this protein's going to come up again. It's going to get a different name. We're going to call it succinate dehydrogenase for now. But what you're going to see is that succinate dehydrogenase, in addition to having this covalently bound FAD group, right, that upon oxidizing that 2 and 3 carbon to make that carbon-carbon double bond, this protein also has a series of iron sulfur clusters built into it. And it's going to take those electrons from FADH2 and it's going to transfer them to that iron sulfur cluster. And then they're subsequently going to roll downhill to help us make ATP eventually, right? So succinate dehydrogenase is the only enzyme that's directly coupled in both the chemistry it's running for the TCA cycle and the subsequent activities of the system that's going to make us ATP, right? All right, so that's reaction 8. We're almost there. Last couple of reactions and then we're done, right? So remember the name of the game here, I keep reminding you guys, the name of the game is we want to get back up to oxaloacetate, right? So we're already down to 4 carbons, right? We had that symmetric dicarboxylic acid succinate, right? Which we just oxidized across the 2,3 carbon to fumarate, right? Now, what we need to do is if you remember the structure of oxaloacetate, remember it's a alpha keto acid, right? All right, it's also a beta keto acid technically. We need to put in a carbonyl somewhere, right? Well, what have we got to work with? We've got a carbon-carbon double bond there. And we got water, right? So in principle, right, you could take some water, you could activate it, you could then take that hydroxyl from the water and you could add it across that double bond, right? Essentially, it's an addition of water across that carbon-carbon double bond to make this molecule, right? Malate. Right? The free energy change for this reaction is pretty favorable. Adding water across that carbon-carbon double bond is, pretty favorable, minus 3.8 kilojoules per mole. Again, I don't know I haven't been able to locate the assumed cellular conditions, but again, it's going to be less than 0. It's going to be pretty favorable as well. Right, so this is part of what helps pull that previous reaction when we make fumarate. this helps us like drain that sink and pull that reaction in the forward direction. The enzyme that runs this reaction, fumarase, helps drive what's already a relatively thermodynamically favored reaction in hydrating that carbon-carbon double bond, okay? One wrinkle here that I sort of didn't mention in the previous reaction, I probably should have, the succinate dehydrogenase reaction. If you think about that carbon-carbon double bond, there are two isomers that bond. You could have the trans isomer, which is what fumarate is, or you could in principle make the sort of the cis isomer here, right, in principle, right? That's kind of ugly, but right, depending upon how you ran that reaction, right, you can imagine making the cis version of that bond versus the trans. The cis version of that 4-carbon dicarboxylic acid with that 2,3 carbon-carbon double bond, that's a real molecule. It's something known as malleate. But succinate dehydrogenase never makes it. It's stereospecific. So it knows only how to make and will only make the trans isomer of that molecule and therefore make fumarate. Likewise, the fumarase, when it acts to create malate, it only creates malate, right? It's a stereospecific addition of that water molecule to that 2-carbon, and it only makes one stereoisomer, right? It doesn't make the other stereoisomer. All right, well, once we got malate, we're almost there, right? Because remember, all we need to do is turn that into a carbonyl ketone and we're done, right? So that's the final reaction, reaction 10. Okay. We're now going to take that malate molecule that we just made. I'll just go ahead and redraw it here for completeness. All right, so here's malate. So now we need to run another oxidation reaction, right? So we're going to take some more NAD, right? We're going to oxidize that alcohol to a carbonyl group to now regenerate our dicarboxylic alpha beta keto acid oxaloacetate, right? Which is cool, plus NADH plus a proton, right? The enzyme that runs this reaction is, of course, malate dehydrogenase. And the free energy change, this guy's not so favorable, right? Malate dehydrogenase would really much rather take oxaloacetate and NADH and create malate. It'd really rather run as a reductase, at least in terms of the thermodynamics of this reaction. That's way more favorable because the standard state free energy change is pretty dadgum unfavorable. It's almost 30 kilojoules per mole biased in the opposite direction. The standard state free energy change of this is going to be less than 0, but probably, again, is just barely. And the reason it is because the citrate synthase reaction, when this reaction, TCA cycle, is running oxidatively, running in this direction, the citrate synthase reaction drains the oxaloacetate pool, as well as there are other things that work in the oxaloacetate pool. That pool is kept so low in general, or so actively drained in general, that this reaction can actually get pulled in the forward direction, despite that pretty unfavorable standard state free energy change, right? It's a theme we keep seeing again and again and again. And why that's the case, or why it's the case, has to do with the nature of these molecules. But it turns out there's a reason, in some sense, that biology has maintained and works on these reactions by pulling them, because it makes them reversible. And we'll see why that's important when we get around to talking about regulation of these pathways, right? And why that's an important thing. Okay, so that brings us to the end of the TCA cycle. So just a last little bit of accounting. to go through here. We're thinking about, we just sort of think about the balance sheet of what we've done as we've come through this. So if you think about, we had 2 pyruvates, right, which gave us, in the end, after 1 round of the TCA cycle, gave us 6 CO2 molecules, right? Which, remember, only 2 of which were from that immediate round of the TCA cycle. The remaining 4 carbons have to treadmill their way through the structure. And so if you'll sit down and think about each of those reactions and where those different carbons go, what you're going to see is those carbons, those 2 carbons that came in from acetyl-CoA, they kind of work their way through the structure of oxaloacetate. The next round, the CO2 group that's left over, it goes. But the carbons in the middle, they sort of treadmill their way down the structure until they eventually turn into CO2 groups and get ejected as well. So if you're not familiar with that, it'd be worth sitting down and just sort of watching the carbons right out the cycle and see what's happening, right? Okay, we also got out of this 8 high energy electron carriers, all right? So we had 6 NADHs net from this 1 turn of the cycle and 2 FADH2s, right? And we got 2 ATPs, right? Or ATP equivalents, right? GTPs, right? So overall then, our total yield for this process would be sort of 1 glucose plus 2 water molecules plus 10 NAD pluses, right? Plus 2 FADs plus 4 ADPs. If we're basically going to fold in the total direct synthetic yield from glycolysis through TCA cycle, so 20 steps of chemical reaction, this would be our net at this point, right? Plus 4 inorganic phosphates would then yield 6 CO2s plus 10 NADHs plus 2 FADH2s plus 6 protons plus 4 ATP. All right, that's been like 4 lectures. Like I said, 20 reactions. And for all of that, what we got was 4 ATP. That's like, really? You know, that was a lot of chemistry and a lot of steps, and I got 4 ATP out of that. Not so awesome for all that work. Well, hopefully you guys remember the payoff is not at the level of these substrate level phosphorylation events, unless, like we've talked about several times in glycolysis, you need speed. Unless you really need to go fast, the energetic payoff hasn't happened yet. The energetic payoff is here. Those NADH molecules and FADH2s carrying those high potential electrons, those unstable, thermodynamically unstable reduced molecules that really want to spit those electrons back out, there's potential energy there that we can couple to. Specifically, each of those NADH molecules is worth 3 ATP, right? And each of those FADH2 molecules is worth 2 ATP each, right? And you add that all up, that means those NADH and FADH molecules are worth 34 ATPs, right? Which is quite the energy yield compared to what you get just from glycolysis and the substrate level phosphorylation of the TCA cycle itself. So that's pay dirt. That's the reason why it's almost an order of magnitude more energy than you can get from, well, it is more than an order of magnitude than what you can get from glycolysis alone. So that explains why this pathway, though it takes an additional 10 steps and more enzymes and is significantly slower to run, it's evolutionarily, selectively really, really worth doing because you really get a maximum yield of energy in terms of utilizable ATP when you run this pathway all the way down to the full combustion of glucose all the way to CO2 and water, right? Okay, so with that, that brings us more or less to the end of TCA cycle. We're now gonna, in the next lecture, we're gonna take a bit of a anabolic break, right? We're gonna see how something gets put back together, specifically glucose, because the way in which glucose can be synthesized is an important aspect of how all this metabolism gets regulated. And one of the reasons it's so important to regulate it is because, as we pointed out, or as I pointed out, with glycolysis, and as we saw in that paper from Luke Cantley, with cancer cells running glycolysis to make intermediates for biomass, TCA cycle, even though it's got this great energy yield, it's super important for making molecules that we need for synthesizing other stuff, right? For example, citrate is an essential component for the formation of fatty acids and sterols, right? Alpha ketoglutarate, man, this is a biggie. Alpha ketoglutarate is key for making glutamate and other amino acids, which are then fed into 1-carbon metabolism for making purines, right? Succinyl-CoA is important for making heme. Oxaloacetate is directly connected, as we're going to see in the paper we're going to read, to formation of aspartate, which is also important for making other amino acids. So the TCA cycle, you run the TCA cycle not only just to make as much ATP as you can, but also to create these intermediates we need for making other stuff. These are the so-called anaplerotic reactions of the TCA cycle. There's a corollary to this, which is we're also going to see, which is that you can feed the TCA cycle. If you don't have a lot of carbohydrate coming in, you can feed the TCA cycle and run it by feeding it in sideways. And one of the main ways you do that is through amino acids going in this way through alpha ketoglutarate, or coming in this way through oxaloacetate, right? Okay, talk to you next time.